Okay, kia ora koutou katoa, ko Black Toka Wingoa, nau mai, haere mai, and welcome. Welcome to our Genomics Aotearoa seminar series. For those of you who don't know me, which is no one in this room, so that's a bit of a strange thing to say, I'm Mick Black, and along with Peter Dearden, I am the co-director of Genomics Aotearoa. It's actually kind of cool that we're doing an in-person seminar and an online one as well. That's one of the special things. The main thing that's special, though, is our speaker today, who is something of an amazing and internationally renowned researcher. And the cool thing is she's actually here with us in Dunedin. So this is not one of those online things where we pipe people in from overseas. We've actually got her right here. So our speaker for today is Dr. Karen Miga. It feels kind of weird being in a room with 6 people and telling her this, but because she already knows. So Karen is an assistant professor at the University of California, Santa Cruz in the Biomolecular Engineering Department, and is also an associate director of the UCSC Genomics Institute. I'm going to tell you a couple of things about Karen. I suspect she will probably tell you a few more in her talk about herself and her research. And she knows what I'm going to say because I'm reading some of the blurb that she sent to us. So in 2019, Karen co-founded the telomere-to-telomere T2T consortium, which hopefully you have heard of. It's an open community-based effort to generate the first complete assembly of the human genome. Some of the more astute of you will have noted Karen is the lead author on the publication that came out in 2022 on the T2T genome. And some more of you will have also noticed her involvement in the recently published work in Nature from the Human Pangenome Project. Where she is, and it's right on the fold of my paper, the director of the Reference Production Center for the Human Pangenome Reference Consortium. Something you may not know about Karen is that for her amazing work, she was also named one of Time Magazine's most influential people of 2022. That was in the innovator category, along with other Kiwi notables such as Taika Waititi. So well done. So that was, this is a fantastic recognition of the amazing work that you are helping drive forward. So well done. Those of you who are online, because everyone here knows why, might be wondering how on earth we got someone like this into our seminar series to travel to New Zealand. And so there I'd like to offer a massive thanks to the GA Variome team led by Steve Robertson and Phil Wilcox. The Variome team has forged a really nice relationship with Karen's team at UCSC, and this visit here with her team is actually part of a reciprocal arrangement that they have developed. And everyone in the room knows, but online you don't. We've actually been having a wonderful workshop on assembling human genomes from long DNA sequence reads that Karen's team delivered in collaboration with the GA training team and the NESI training team. So it's been this massive team initiative. It's been wonderful. I managed to attend about half of the sessions. Probably one of the most useful workshops that I have been to in a long time. Sorry, Tyler, we've written a lot of workshops. But I think you'll forgive me on this one. So I feel really lucky to have been able to be involved in that. So thank you so much for bringing the team over. Everyone's waiting for me to shut up and for you to start. So I'm going to invite you up here to tell us about some of the wonderful things you've been doing. So thank you very much.
Thank you very much. That was an incredible introduction. I actually did not include the Time 100 thing in my write-up, but it is kind of interesting because when we had our Time 100 dinner, we were actually sitting right across from Taika. So it was kind of bringing worlds together once again, I guess. Once again, thank you very much for the opportunity to come to beautiful Aotearoa and share in this wonderful workshop as just discussed. This has been kind of a gift for our team to even work at this level of training this material for the very first time. And I feel like this is just a wonderful way to end this time here with our collaborators. So let me get started. I'm here to talk about reference assemblies. This is something like the ground we walk on. Many geneticists and genomicists often take for granted how far we've come because we have access to a human reference genome. I use this in the workshop as well, but I always like to make people not think of it as a series of A, T, C's, and G's, but rather a file that contains such rich information that can be downloaded anywhere in the world. And what it's done over the past 2 decades is it's kind of driven research in terms of basic research, clinical genomics, as well as understanding human histories. Now we are bridging this 20-year, the 20-year anniversary of celebrating the first release of the complete human genome, in air quotes, where here we're actually celebrating the release of euchromatic sequence. Everyone in the scientific community knew at that time, back in 2003, that it was just a portion of the genome, not roughly 92%. And there were 200 million bases roughly missing from the human genome. Now, if you were to kind of zoom into the reference genome at this time, what you would see is a string of characters like ends that were more of a gap or a placeholder. And we knew where these gaps were. They were predominantly in areas that were known to be repeat rich, like human centromeres, gaps in the arms, which we knew had gene duplications, subtelomeric regions, as well as entire short arms. And this is really taking a figure from the human genome paper to really illustrate this, because your eyes can begin to capture all of these big red blocks where we know the centromeres are located, and all of the ticks to show you where the various components of the repetitive genome were that were missing before. Now, this is my opportunity as a repeat biologist and how I got into all of this, to tell you that these repeats aren't just a way for us to close these gaps. It's not just kind of topping up, but rather these are elements that are fundamentally important to life. We know that these centromeres, for example, are fundamental genetic units that are important for proper chromosome segregation every single time our cells divide. We know that the ribosomal DNAs that are embedded in each acrocentric short arm are important for instructing, our cells on how to make proteins by constructing the ribosome. And also we know that even the regions that present repeats within the arms of the chromosomes are often sites for genome instability and expansion and contraction of gene families, which have a role in cellular function. I also love showing this picture of what appears to be a chocolate chip cookie. This is not a human cell, this is a mouse cell, but the densely staining sites that I'm showing you are visual regions of condensed constitutive heterochromatin. These repeats that we're talking about in the acrocentric short arms and the subtelomeres and the centromeres, that's what lives in these spaces of your interphase nuclei. And these are regions that are highly specialized. They have unique chromosome structure, function, they're late replicating, they have unique mechanisms for DNA repair. We understand from kind of low resolution IF studies that they have unique chromatin organization. We have only scratched the surface in terms of understanding RNA biology, how these regions respond to viral integration and response. And it's amazing how wonderful these regions are in terms of their dynamic states of evolution and how much more we have to learn. So I'm standing in front of you because we're celebrating. We're celebrating the release of the first complete human genome. As I mentioned, this is, roughly 200 million bases relative to the previous reference, which was HG38. And this is the team here. This is a meeting. These are the two youngest teams and my daughters who I pulled into the TAT inadvertently because we hosted this entire consortium during a pandemic where most of us had our kids at home with us. This is the first time I met many of the colleagues at the T2T consortium face to face in Santa Cruz because we were an international grassroots effort. And I love this picture. Oh, okay, great. No, it's fun. And I love this picture too of the cover of Science, not only because it really illustrates these bright red bands I introduced at the beginning of my talk of these blocks of DNA that were missing. However, it's not just a celebration of closing the gaps and not only a celebration of the people who were involved, it really is a celebration of technology, which brought us here to New Zealand as well. And that's using a combination, kind of a technical achievement of both HiFi, which is the PacBio platform, which I'll talk about a bit, and DNA sequencing from the Ultralong platform from Oxford Nanopore Technologies. And I really love to show this slide, not only to illustrate the two different long read technologies that were critical for spanning a lot of the existing gaps, but to really also credit Sergey Nook, who's a postdoc in Adam Phillippe's team at the time, who is the lead author of the complete genome paper, who sadly was not at the table with us at time 100, but should be because he came up with a lot of the innovative methods that allowed us to reach the finish. No, stop. Also, I love to also remind audiences that it's not enough to assemble a genome. I was kind of tickled throughout the workshop hearing over and over again my dear colleague Adam Phillippe being quoted of saying an assembly is a hypothesis, and so it's my turn to do this on stage too. And it's a hypothesis that needs to be tested by quality and assessments. And I love the Anne's in the audience because she was really driving a lot of the methodology, as well as Laurent Rie and a lot of people in our group to really understand how close we can get to perfection. And in doing that, we celebrated in that Science cover all of the different chromosomes, and that even includes the most gnarly chromosome. Those of you who love human chromosomes, that's chromosome Y, where we can not only get into the most ampliconic regions that we know contain genes, but millions and millions of bases that I'm showing in this beautiful stained glass plot. This software is developed by Mitchell Volger, who's also a student in our T consortium, which really illustrates these massive tandem repeats that were never seen before this type. So now that the human genome is finally complete, what can we learn from it? I think that's always really important. It's kind of a nice technical exercise, but we're doing this for a reason, because as human geneticists and genomicists, we want to go back to that first map where we're all downloading it and we're starting to learn about basic clinical and human histories again. And so to do that, we took a couple of steps. The first one that I'll go through briefly is this idea that you can take short read data, which is what most researchers typically use when they're looking at large cohorts. And we used data from 1,000 genomes to map back to CHM13. CHM13 here is in orange, and HD38 was the prior assemblies in blue. And what you can hopefully see from the slide is that you see a kind of lowering of CHM13. And I'm just using this to share that all of our mapping statistics really showed an improvement of short read mapping when performed to a more complete genome relative than before. This means that you had less low quality reads, less Mendelian violations, and overall we were discovering hundreds of thousands of novel variants, detecting and identifying tens of thousands of error-corrected samples per each sample, 1,000 genomes mapped. And this is really important when you start thinking about clinical genetics and genomics, because we found a 12-fold reduction in these false positive calls in medically relevant genes. Much in tone with the workshop that we gave, all of the tools that we tried to develop, we put them into workflows and put them on ANBO so that other researchers could use them as well. Another really cool thing is that we know there's copy number variation. This is a kind of a larger assessment of maybe genes that are multi-copy. And when we started to evaluate copy number variation across panels of human diversity from the Simon's Diversity Project, we found that, in fact, CHM13 matched copy number estimates broadly across all of this sample diversity through these cohorts than HD38. And the reason for that is because we finally had with CHM13 a more exhaustive or complete look of all the gene families that exist in a human genome. This is celebrated work from Mitchell Vogler, who's in Evan Eichler's group, who's dedicated, Evan, that is his career to segmental duplication and discovery, and now Mitchell's taking that torch and leading his own research to try to expand these maps even further. And as you can see here in red, these are all genes that were not present in the past reference genome that are now present in CHM13, and the ones in black were shared with HD38, but they were in a different configuration that made them prone to map as efficiently. And this is really important because this really addresses the need of this T2T genome to address completeness bias. Let's say, for example, you had two genes. I'm trying to illustrate them here. One's gold and one's yellow. The yellow one has a mutation. And if you were to just generate short sequence reads from these data, a portion of these reads would look identical to one another. The only way you can really distinguish them are these smaller ones that contain or span the mutation. And if you had a genome which was collapsed or didn't represent the true two copies, but rather only one copy, all of that information would be piled onto the one representative copy, and then you can call false variants. And this is exactly the type of information we were seeing when we were starting to map short read data to HD38, as I'm showing you here in this kind of extremely colorful map where you're seeing this pileup of variants, where we know there's 9 copies of a gene, versus the true 23 copies that we observe in CHM13, where now all of a sudden the alignments are finding their true match. I really emphasize, as someone who loves repeats, I usually don't spend so much time in any of my talks talking about genes. And so that was really for the audience's benefit more than my internal research joy. Mainly, I love thinking about these incredibly difficult regions that are full of highly repetitive sequences that typically require long read sequences to really get into these regions. This is just one slide to let me walk you through the ideas that you can now not only get the acrocentric short arms and have complete sequence all the way across, but now we're building detailed maps of the repeat annotation, gene annotation, whether or not the genes have methylated bases, whether or not they're expressed by looking at ProSeq or IsoSeq or all these different sequencing technologies to look at transcripts in these regions. And we can even compare between the different acrocentric chromosomes to begin to look at their similarity. What I'm hoping you take away from this is that we're not just walking in with a sequence map, but rather a rich annotated map, and we can begin to start to study interesting biology. One interesting biological question is thinking deeply about Robertsonian translocation. This is a very common structural chromosomal abnormality seen in the general population that has an incidence of about 1 in 1,000 births. we know that even though all the acrocentric chromosomes can go through this type of breakage and fusion, we see it predominantly happening in 3 chromosomes. It's 13, 14, and 21. So it's really fun when you start to have these really detailed maps of all the acrocentric short arms for the first time. You can begin to kind of drill in and understand not only the satellite structure that I'm showing you here and all the patterns in the genes, but then trends that are actually shared between particular chromosomes. Here I'm just identifying, for example, a block of homologous sequence that spans both satellites and segmental duplication in genes on chromosomes 13, 14, and 21 that are near identical to one another, yet have opposing orientation that could lead to new mechanisms of how these types of events could happen. Now, my whole career, of course, has been focused on satellite DNAs. These are the tandem repeats that are on steroids that typically are found in centromeric regions in the human genome, where you find them in this kind of head to tail forever, but millions of bases, and they're pelted with these segmental duplications in tandem repeat classes. I love to have a little bit of a chest thump moment here where I say, of the CHM13 release, the vast majority in this kind of Pac-Man appearing plot are this, what we call synsats, or the centromeric associated satellite DNAs that were missing from the previous reference genome. So we now have high resolution maps of every single human centromeric region. I never thought in my entire career I'd be able to say that so easily on just one slide. But each of these regions now have detailed base level maps of regions that we know have kinetochore assembly are important for chromosome function. Understanding precisely where the kinetochore comes in that actually says you're the genetic site where the centromere works was something that had never been done before. No one really knew. We had assessments that were kind of roughly guessing where centromeres were positioned, but we went a step further this time and said, hey, can we begin to study the epigenetics of the centromeric regions to predict the sites of kinetochore assembly? And the way that we did that is that we actually looked at signatures of CpG methylation. The wonderful thing about long read sequencing is I'm trying to illustrate with one platform. This is Oxford Nanopore. As you start to read these long sequences through the pore, you can begin with high accuracy and precision, distinguish between CpG and non-CpG methylated bases. And when you take these long reads and precisely map them across centromeres, you can begin to see, as I'm showing on this kind of squiggle plot, as you start to enter into an alpha satellite array, you can find these very clear drops or centromere dip regions or CDRs that we know map to regions where the kinetochore is assembly. Of course, one human centromeric region is not enough. We started mapping all of these different regions because we understand that if you had a bigger region, a smaller region, maybe it's misplaced, that could actually lead to something called aneuploidy, or where when the chromosome divides, maybe you could have more or less chromosomes, and that can lead to cancer or various, implications in health and early development. So we began to study this in earnest. We've been building a number of these T2T genomes now and trying to think through how we can look at CDR placement and variability. So we're starting to build maps not only of the DNA sequence and how it varies, but actually the position and the span and the structure of these centromeric regions in terms of the epigenetic information too. And here I'm just trying to show you a panel of X chromosomes where we're looking at the X centromere only within the span of each of these windows. And I'm trying to illustrate that in the middle and the top, we have a grouping of centromere positioning that happens in the center of an array. In the middle, we find groupings where they're found on the Q proximal and then also on the P proximal regions. And we can map this, of course, not only using long-read sequencing from Oxford Nanopore, but also now from the HiFi platform using Primrose. And so this is another kind of question that we're actively pursuing in my group is, what does this type of new variation, not only in the sequence level where you might have a very small array and a very big array, but also the epigenetic positioning of where that functional component is that makes a centromere. So building up kind of a sampling or a varium of our own, thinking about where these satellites and epigenetic variants are really important to some themes in my group. Now, before I transition, I did want to bring in that we didn't know all the repeats when we started. When we started to assemble these genomes, much like we were talking about in the workshop, started to build annotation on top, we were finding massive regions on the order of 100 kilobases that were just blank. There was no information there for our team in terms of the repeat content that was there or gene content. And so that really took in a whole other aspect of the T2T group to do new repeat modeling and repeat discovery. And I really want to credit Rachel O'Neill and her student Savannah Hoyt, who led this work. And so in this case, what we were doing is we were predicting new repeat elements that were not present in HG38, and we reported 5.5 megabases of these new repeat predictions. Now, some of the newer repeats weren't actually new repeats at all, but new combination of repeats. It was almost like what I'm trying to illustrate here for the acro-satellite tandem is that you had a combination of different types of repeat elements, and then those combination or composite of repeats would then be repeated over and over and over again. This is similar down here for the beta composite array. And we were starting to find these types of unique composite or Frankenstein tandem repeats throughout our different karyotypes of different chromosomes. And this, I think, is really cool because it illustrates 2 points. One is not only did we have a part of the genome that we had no context of before, so new discovery at the genetic level, but we're also able to do the same trick that I was showing you with the CDR, where we can begin to study epigenetic patterning. And here, doing the same thing where we're looking at CpG methylation by looking at the alignments of reads like Oxford Nanopore, you can see these very diagnostic depths like the CDR once again. Where here I want your eyes to focus on the blue spots separated by both reds. And we have no idea what's happening in these types of regions, but it's once again hypothesis driven research to where one can go in and begin to study this in earnest. And so once we got done with this work, I was really thinking this would go straight to healthcare. Hoping that people in the clinical space and clinical genomics would start to begin to mine for new variants, hidden heritability, start to look at these satellite DNAs and centromere structures that may be driving some of the phenotypes that hadn't been explored or even studied in earnest before by having a more complete genome. I think at the same time, a lot of us were really motivated in expanding the use of the tools and the methods that we use for human into other non-human spaces. So that launched a number of T2T genomes, may it be T2T mouse, rat, zebrafish, fly, non-human primates, T2T ruminants. It seems like our Slack channel has just become this buzzing activity of communities of interested researchers from all non-human spaces coming together, trying to figure out ways to look at comparative genetics and genomics. And of course, I think, as I mentioned my chocolate chip cookie slide, there's real information here in terms of discovering new cell biology. And I think that now that we have complete genomes, there's decades of work here to really figure out how these regions that had been missing or ignored for decades can tell us more about who we are and how our cells operate. But one genome is clearly not enough. And the reason for that is at the end of that chocolate chip cookie slide, I had that evolution bit, right? And that's because we know that a lot of the sequences that are being introduced through these T2T genomes are highly repetitive, and they're also very dynamic. You can see this in the case of human centromeres, where once again, I'm trying to bring your eye to the size, where in chromosome 2, you can have a very small array to a very big array, and we can begin to measure the size distribution of these types of assembled arrays through panels of humans. We also know when we look at gene copy numbers that this can vary quite a bit. If you were just looking, for example, at a gene SMN, and you're looking in the original human genome, you would estimate that there's 4 copies. If you're looking in CHM13, you would also see 4 copies. However, if you're going outside of these 2 genomes, you would begin to see in the diversity panel that many individuals around the world happen to have, 4 copies, 2 copies, 6 copies. And it really is ensuring that we have adequate copy number differences that are within the human population properly represented. And then even the acrocentric short arms, where I was telling you the story about 13, 14, and 21, now there's been some new and exciting pioneering work that would show that some of these shared spaces are actually happening not only once or twice, but this might be natural variation that's happening within the human population, where there's arm switching and some rearrangements that are happening within the space of the acrocentric short arms that's at the chromosome level that we just haven't been able to see before. And it's not just the repeats. Even as you walk across our genomes and the seemingly unique and easy to map regions, we've known for many years that there are structural variants. These are bigger variants. These aren't single nucleotides. These are, on the order sometimes of 6 kilobases in length that are inserted or deleted that differentiate my genome from everyone in this room. And then we understand as well that there's tens of megabases of common polymorphic sequences that are missing from any one individual reference. We know this causes reference biases in terms of mapping. And if you had a cohort, for example, where you had a catalog of known large variants, and you were to take the short reads generated from that catalog and begin to map them to H238 using our standard protocols, we've estimated that about 70% of these big events are missed by all of these types of assays. And finally, I think a lot of researchers that I work with start with this slide because I think that this is really incredibly meaningful as well, in the sense that it's not just about capturing sequence, may it be the dynamic complex sequences, may it be the structural variants, but it's really about representation and who's represented when we perform these genetic research questions in the future, and how that could actually lead to new biotechnology and health inequities. So that allows me to pivot, because I think the T2T consortium is brought into the mothership of the Human Pangenome Reference Consortium. And it's not a different goal per se, in the sense that we're still incredibly invested to understanding how to use long read and innovative sequencing technologies to look at more complete, more accurate genomes. However, instead of moving on a celebration of a complete genome, one that we know represents a single European haplotype, perhaps we can start to take down that next technological barrier and start thinking about how we can expand these tools and production scale to reach hundreds, if not thousands, of T2T diploid genomes in the future. And this allows me to switch my hat in front of you now and start to think through the goals of the Human Pangenome Reference Consortium. The Human Pangenome Reference Consortium really wants to reboot, if I could, the idea of not HG38, not CHM13, but it wants to break the wheel of our dependency on one linear genome for references, period. And the idea behind that has had a long history in pangenomics, celebrated literature in microbial genomic spaces as well as yeast spaces. And I feel like it's getting a new day in human and higher eukaryotes. And what it means is that you need to have a sampling or a representation of a population for that to be meaningful. And it also means, at the moment when we started this, it's a big effort to get this off the ground. You saw the picture of all the people who leaned into the T2T consortium. Now think about doing that for hundreds and hundreds of genomes at scale. And in order to do that, we had to kind of work with this highly collaborative, multidisciplinary, cross-institutional working groups. It sounds like I'm writing a grant right in front of you, but you know, it takes all of these different working groups working seamlessly together for this to work. And we all work with humans, and humans are hard to work with. So the idea that I can say, you know, this works, I think is a triumph all of itself. The idea that, you know, there's a team of research scientists that are thinking deeply about how we're performing population sampling and representation within the context of this project. There's a team of scientists that are working on the edge of company partnerships to ensure that as soon as new technology comes out, we're testing it, we're optimizing it, we're onboarding it. And then we also have a team of recognized scholars who have been developing innovative methods for T2T assembly, who we're so grateful to have as part of our team. So as soon as we take in new technology, it's kind of a back and forth and a partnership that's churning in this area. And then, of course, it's not enough to have reference assemblies. You have to understand how to utilize those types of reference alignments in a meaningful way or in a pangenome. And so we've organized a team of experts that now are working on that, I guess, I want to say the side end, but you know, this beside the assembly experts, and trying to incorporate that rich information into a resource that's useful. And then I've recently started to, maybe I'll just keep building my slide out as everyone starts to meet with me, but I've recently changed it to really emphasize this new component where it's been such a domestic effort. We've been largely organized within the United States for the first phase of our grant, but I really think that we're making strong inways with trying to become more of an international project, playing once again to the tones of the original Human Genome Project, which means that, and I'll touch on this as well, is reaching out and becoming more of a federated effort. And I never want to leave this slide without emphasizing along the bottom is that we organized this really with ELSI scholars who taught us the importance of embedded ethics and policy. And by doing so, what we have benefited from is having ELSI leadership join each of these different working groups and providing guidance as our group move forward. So you might ask yourself, as many people ask me directly, who is currently included in this pangenome? And so at the beginning of our work, we decided to focus exclusively on cell lines that were available to us through a previous effort known as the 1,000 Genomes Consortium. There are many reasons for this. One is that the individuals who participated in the study, they did so because they wanted to have a representation of normal variation at different points around the globe, so that we know we have 3,000 individuals. We all know the population of the Earth, 3,000, a very small number, but this was meant to be a sampling from outside of the United States. Also, the individuals who participated in this study, they consented for open data sharing, and that was critical to the success of this work because we wanted to ensure everyone around the world could download this pangenome and could benefit from it. We also wanted to have access to short read data. All of these genomes already had that, so that we could do a little bit of pop-gen in terms of justification of who we were to include in the study. And we had the benefit of existing lymphoblastoid cell lines. Now, this one's kind of something I don't think a lot of researchers pick up on, but as someone who's been thinking about the genome for a very long time, it was a bit of a struggle not having an associated cell line with HG38. In the sense that a lot of the discoveries that need to be made, to go into the cellular context, it can really advance some of the biomedical questions of basic science and cellular function. Now, something also I want to point out before I leave this slide is that our goal is a little bit different than a lot of other consortiums that are trying to understand disease association by digging deeply into rare alleles or dig deeply into sequence information from those of disease cohorts. Rather, our goal is to represent common human haplotypes with an allele frequency of about 1% of the global population. And so in doing that, our goals, of course, are to cover genetic and geographic diversity, allow for these low passage cell lines. And at the beginning, we were really beholden to trying to keep parental data in order to enable phasing or the use of trios. There's something that I thought was really important that I think was motivated through a series of conversations with our LC team and our population sampling representation team, was the idea of removing these population labels that were part of the 1,000 Genomes Project and really looking at the common variant spectrum in order to make these justifications. Of course, our team fully understands that the 1,000 genomes is insufficient to reach the level of variant discovery and representation that we need for a project this ambitious. And so we've teamed up for our next step with Emer Kinney, who's the PI at the BioMe Biobank, really taking advantage of the rich diversity in the metropolitan area of New York City. Here we have over 70,000 participants with sequencing data. So we can go through once again, work with our LC scholars and our population sampling and representation team to begin to identify individuals who may present common variation that's missing from 1,000 genomes and go through recall and reconsent. We also are exploring recruitment efforts through Washington and St. Louis, in the St. Louis area, where they've built a community outreach program for African Americans who live in the area. Once again, this requires a partnership with our LC team for consent and outreach, where we've had to think deeply about how to work through these outreach materials, work with governance boards, and also have review and use of an external IRB. Okay. And so in this call to action, of course, there's a lot of emphasis that I just went through thinking about who is part of the pangenome. And then at the beginning of my talk, I went through all of the beauty of what it means to have a T2T genome. And that's what we're going for. We're trying to build the most rich information from each individual to build a database of global genomic diversity. And for the first phase of our grant, what that really meant was aiming for at least 350 diverse T2T diploid references. And you can think about these references as just being that, a multiple alignment of all of these different human haplotypes, as I'm trying to show you in this cartoon. And if you were to zoom in, you can actually organize this information in a very intuitive way, where all the haplotypes, the areas where they're shared, could be grouped together in something called a node, and then you can have the edges distribute where, or distinguish where they're different. Here, for example, I'm showing a copy number variant, a single nucleotide variant, or an inversion. And so we've been making these quantum leaps in de novo assembly, not only for the T2T genome that we talked about, but really at this production scale of trying to scale up to hundreds of genomes. And I like to show this slide of one genome because I want people to realize that this T2T genome, we're always talking about one chromosome one. HG38, we're always talking about one chromosome one, but everyone in the audience and hopefully online as well understands that our cells, our somatic cells, are diploid cells. So we actually have two genomes, and we haven't been able to to phase these two complete genomes and create that same level of accuracy on both phased haplotypes until only recently. This is a very exciting time. And so this is what I'm trying to show you with the blue versus red, where now the red is the maternal genome and the blue is the paternal genome. And I'm also wanting to illustrate at the beginning that we still have work to do, what you're seeing with your eyes in gray, where we can automate this process, but there's still a human element to take it to T to CHM13 level. We've been working with two amazing scientists in this space, of course, Adam Philippi, who I've already credited with a lot of the leadership from the T to T efforts, as well as some of the critical assembly algorithms that were important to take it to a complete chromosome assembly. I also want to credit Hung Lee, who's been driving a lot of technology as well. And we're really at this point practicing both. And in the workshop, we tried to introduce students to both of these assembly methods. But the ideas are still using the same, dare I say, long read production protocol, where you're operating off of a mid-length, extremely high accurate sequence like HiFi, where you can build that first string graph that we talked about in class. And then you can use these very long sequences that are generated from the Oxford Nanopore platform to kind of thread through that graph and resolve some of the tangles. And then when we were talking about phasing, that picture I showed before where you have red versus blue, or knowing if that genome is maternal or paternally derived, you can begin to use information like the parental data or Hi-C, and that's something we talked about as well, to reach these types of finalized contig information. And in doing this, what we were able to reach, and I used the we in a royal we sense and really want to credit the people on the slide for the actual achievement, is the first T to T diploid assembly. And this is using a cell line that was high value of the Genome in a Bottle consortium known as HG002. And now we have complete maps of every human chromosome. Because I adore Adam so much, I'll tease him because he's not here. He's a perfectionist. So of course, the next thing he wanted to do is take this genome to QV100, which is like as perfect as one can get. So it's an incredibly ambitious goal. And it's something that I think an amazing number of talented scientists within the T to T consortium have their head down working on now. Now, why is it important to be perfectionist like my colleague Adam? I would say that the assemblies that we've been releasing before we started moving to these new protocols are pretty fantastic. We were already seeing phasing, right? But there are important clinically relevant genes and complex sites in the genomes that could benefit clinicians to have a better, more accurate map. One example you might consider is SMA, where we know that 1 in 50 are carriers. That's a terrible disease for babies. Like, look at Stephen, who probably has the world's knowledge of all of this information because it's one of the leading causes of early infant death. And it's very difficult to study these regions with short reads because it's so complex. We know that the two genes SMN1 and its paralog SMN2, they reside in a highly complex genomic region on chromosome 5, and sometimes even crossing over a conversion between these two paralogs lead to variable copy number between the two. Now, before we started using methodology like Verco or HiFi ASM with ONT, we would get assemblies. They look beautiful, but when we would look at these very important medically relevant regions of the genome, you would see these breaks. And here are these black, that's Karen Miga, making it more pronounced. But there are breaks in these assemblies where we just couldn't make it across. And once we started to utilize some of these more appropriate integration of the long read sequences with HiFi and ONT using Verco or HiFi as ONT, we started to see that we could confidently make it through all of these regions, and having complete comprehensive studies to these very important medical regions is what we're trying to do. And we've also been working with some pretty incredible research scientists who have helped us visualize this. This is just looking at a pedigree that I've been assembling within my group, really crediting Monica Chukova, who's a postdoc in my team, where we're starting to look at grandparents and mothers and the offspring to see how that transmission of information of these complex sites can be transmitted. Okay. And I have a couple more slides before I kind of transition again, but I did want to credit the first release of the human pangenome that really has been building off of that crazy chart I showed you at the beginning of this section. All of the work, all of the people really leads to this one release of how do we put these assemblies together? And so our first draft release, which is in the title, this is just the beginning, was an alignment of 94 of these high quality phased haplotypes. And in doing so, what we're looking at is kind of a wonderful gain here in terms of sequence information that's new. We had 120 megabases of new sequences that we added to the reference, greater than 1,500 of gene duplicates, and then we have greater than 20 million of small variants. And in total, when we were talking before about the importance of these larger structural variants, we can now say that we assessed about 70,000 of these structural variant sites within the context of this very small number of genomes. And as Benedict mentioned during the workshop, the exciting part of this is really giving access to complex regions for the first time. I think everyone is typically aware of sites like HLA, which are important for antigen, for organ matching, or even understanding kind of response in our immune system. And here we can begin to not only map the variants that are in each space, but begin to understand their proportion in different individuals. So this is where it seems really exciting, but from the inside, I can tell you it's pretty terrifying, because we have a long way to go in terms of expanding tools and ensuring access to researchers. And we understand that adoption is going to take some time. No one in their right mind thought that this would be quick and easy, right? But so we're really taking seriously in this next phase is kind of, the drumbeat of improving mappers, downstream tools, making this as accessible as possible. And it's incredibly fast moving right now and exciting. We've already started a little bit of this in terms of tool development. We have a lot of exciting methods such as giraffe and deep variant and Pangeni. I'm putting all of these citations because these are published work outside of the pangenome, just tools that we were utilizing and demonstrating the strengths within the context of a pangenome. We've been working with collaborators now to ensure we get proper representation of tandem repeats. These are things that are commonly missed in short read assays. We understand the importance of looking at RNA sequences in the context of pangenomes, so we're doing more RNA-seq tooling around pangenomics. And of course, looking at ChIP-seqs and other ways to do functional assays is something that we're taking into account. Now, in the past, before this amazing workshop, we mainly were working through conferences where we would show up, we would create kind of content, we'd make recordings, put them on YouTube, and then we'd make sure we'd host all of our information through DocStore, various cloud platforms, Anvil, and then also GitHub. Okay, and that allows me to do a little bit of a pivot because that was phase one. And I feel like, if I can speak honest to everyone in this audience, where my head typically has been for the past 2 weeks is phase two. We're right in the middle of writing a lot of our grants right now. And we have a lot of big challenges ahead of us. And I think one of the biggest challenges is thinking about how we can move into an international space. And so when we were thinking about the Human Pangenome Project, we instantly were thinking, how can we align around global organizations that are already celebrated for organizing, members of the research community across the globe, like GA4GH. So we're starting to put in applications for driver projects, starting to work into shared working groups, and we're starting to build collaborators around the world, thinking about how we can organize new technical standards, how we can think about alignment with ethical policy standards and responsibility, really, for securing resource sharing. This all sounds great. And it should be pretty straightforward. And I think that, if we just took it at that GA4GH knows everything, we're just going to kind of lean into it. Maybe that's what it will end up being. And maybe one of these days we'll have this wonderful thing where this pangenome kind of can be downloaded and it can be the driver, once again, of the baseline of all of genetics and genomics. However, I lie awake at night because I worry about the legacy of our work. There's been a lot of past mistakes other genetic and genomic studies have performed. And I really think that we have an opportunity here to be more innovative, more thoughtful, and really have questions more than answers drive our way forward. And so things that when I think about programs I'm most excited about driving our team, it's, driven under questions of who's benefiting when we make these international relationships, which human haplotypes are actually being represented in this resource. If there's a person who's going to a clinic anywhere in the world, can they benefit from our reference resource? That should be driving our population genetics teams. Inclusion, even the researchers, who are we including? Who are the stakeholders? How are they contributing to this goal, and how are they benefiting from the goal? These are two that are important. Not only inclusion and benefit, but there's an element of justice that I think needs to be really capitalized in our future work. Who will gain financially? Who will gain through knowledge? How can we start to just monitor this as we move forward? And then access, who will have first access is really an important question. I don't think I've really read a lot of bioethics and kind of contemplation of like just the tooling, these workshops. How do you begin to communicate new discoveries and how do you get to benefit from it? And I think this one is something, fortunately, that does get a little bit more attention, and it's at what cost? Who can be harmed by this project? And how can we avoid repeating historical missteps, which hopefully we're all starting to become well versed in? And how can we, as a consortium, become innovative to meet these challenges of today? So with that in mind, I have to admit that I didn't have answers to any of this. And I've been learning organically through trusted relationships and shared commitments with collaborators. Things that I've been learning is that maybe we can establish international partnerships with different models of data sovereignty. What would that look like? How can we structure ideas around that? Can we establish meaningful partnerships that recognize, ensure, and respect non-Westernized ethical frameworks, which is something that I think is really an exciting space to think into? Can we strengthen these partnerships with formalized bidirectional knowledge exchange? Can that be part of the way that we engage in the future? And in doing so, can we structure our goals? Just like I mentioned on the slide before this, everything we do, developing methods, tooling, partnership, communication, with the idea of what's the outcome? How are we going to end this collaboration in terms of benefit sharing models, and what does it all mean? Now, of course, I'm here because of a wonderful group of icons that I'm showing on the front, a bunch of logos that typically decorate my slide, and I'm so grateful for the teams of Genomics Aotearoa, as well as the T2T and the human pangenome work, which is really, I'm just a talking head for a huge number of talent there, as I mentioned. But I also think I'm here because of 4 individuals who formed a true relationship. And I think that that's incredibly important in moving and learning organically in this space. And I think the way that it was so natural that this type of collaboration took place wasn't because anyone stepped up and said, I would love to understand how to make international collaborations in the future kind of broadly cast. It was through conversations where there was a clear focus on a benefit of a collaboration that could have healthcare kind of on the radar. How can we use new technology, T2T genomes, to expand studies of unique variation within Aotearoa populations? How can this effort support and promote accurate diagnosis and make this part of healthcare initiatives here and useful for the population communities here? And I think that's really wonderful because it was also at the same time paired with concepts of Tikanga framework, the idea that it's critical that this project is actually Maori-led, that it's co-developed with processes that benefit of guardianship and thinking about data in alignment with ethical values. So we were so lucky during that conversation, the very first one, that Phil and I actually found that we had more in common than just our interest in genetic variation and perhaps technology, but a passion for education and as well for teaching ethics. So as I mentioned, I was a satellite DNA biologist when I first stepped on the scene. I did not know very much other than Gila and a couple of historical stories, so to speak, through my ethical training. However, being part of the human pangenome kept me up many nights and got me reading many books. And it turned out when I was teaching my human genome courses on the campus of UC Santa Cruz, every course had a bioethics or global ethics component to it. And so when all of a sudden we had an opening for our bioethics class in our BME department, although I felt woefully underqualified, I stepped into that space and said, let me do this. I want to ensure that the students on our campus, when they leave, have robust training, not only in sequencing technology and thinking about, cool tech trends, but can really put that in the context of the social importance of justice, ethical, social and legal implications of the work. And so in doing that, I was really impassioned in taking this role, and I had signed up for a class known as Bioethics in the 21st Century, Science, Business, and Society. And lucky for me, just casually talking with Phil during our first meeting, he mentioned that he also has a passion for teaching ethics. And he shared with me on that call just a slide deck, which I started to instantly think about how can I start to weave this into some of my class. And so when we had Phil on our campus earlier, it was really kind of selfishly a wonderful opportunity for me to really learn on how to improve our coursework. And in doing so, one of the first classes that I teach is on moral reasoning. And it's something I get a lot of positive feedback from students, because what I try to do is I try to, of course, go through the standard normative ethical models that go through standard Westernized thought, but then I go into a whole non-Westernized component of the course training. And I'm delighted to be able to bring in some traditional knowledge systems into the coursework for our students. I also really appreciated in the talk that was given by Phil that there were really some practical applications and thinking about real world ways to apply some of this knowledge. And one of the examples they gave during the talk was how to kind of do a group review of genomic research projects. And in my class, what we've been doing is peer review of different students' work. And we've been thinking about how to do this in the context of genome editing, AI ethics, and even human subject research. And so there were a lot of opportunities, I think, for real growth and curriculum development in that space. And I really think this was something that coming on campus and having these constant conversations back and forth is so beautiful, because it's not like it's just, I'm going to change my notes in class. There are all kinds of wonderful opportunities to think about in the future about how to even bridge more programs. I'm designing a minor on campus for science and justice, and it's an opportunity for students maybe to do cross training between different universities and pick this up in a more robust and earnest way. And then on the other side of that bridged or bi-directional knowledge exchanges that we just talked about is this technical workshop. And this, has just been wonderful. I almost feel a little guilty because I think that we benefited just as much from putting this workshop on. It's been a true delight. It's really taken our team a huge amount of appreciation of all of the infrastructure and the organization that goes into these workshops. And we've never designed anything like this before. Everything that I talked about at the beginning of my talk, that's all brand new. Not many scientists around the world are doing it. We've never taught this to another group. And I feel like expanding this type of information and building this type of course content goes and ripples outside of this short 3-day workshop, because the materials I hope we developed are still useful outside of our visit, and the organization, all the structure, I hope that we can continue to work with the team, because it seems like it's incredibly useful as we start to meet some of the demands of doing additional outreach. This is a picture I took today because of Stephen. So super host. I just want to, of course, say thank you very much, but it doesn't stop there. I think that's this kind of organic learning process, right? I think Anne and I have benefited from having these types of conversation of what does this collaboration even look like? And it really has been thinking broadly and creatively about what is the end game here. Is the end game, like I said before, it's not just learning how to build genome assemblies. It's not how to put on outreach. It's not, it really is a broader goal of how do we start to actually bring benefit to the communities and the participants that contribute into these studies. And how can we do this by building new innovative genomic methods? How can we improve tools to ensure that they can be incorporated more fully and appreciated in healthcare? And how can we do this again with the lens of ethics-driven protocol and training? And of course, here I am once again emphasizing in this relationship the importance of having a dominant ethical model that is done through this collaboration. But of course, I think even building out from New Zealand, this should be something that we template, where we start to have an ethical-driven model with every approach that we do with communities. And I show this picture because it's so stunningly beautiful here that it's so sad for me to leave. But the nice thing is that this once again gets carried into our own space. So this isn't the end of our collaboration. This is actually once again another step toward a new beginning, where we're going to welcome David and Ben and hopefully Phil again to UC Santa Cruz's campus. This is us showing off our big, beautiful trees, as my daughter likes to call them. Once again, this is building into the collaboration of how can we work together to build new innovative tools in this space. So with that, I will end with my standard slide of all of the people's faces who I have the pleasure of working with at the Pangenome and the T2T Consortium. Along the top are all of the contributors to the first phase of our project. And on the bottom really are the company partnerships, who I don't think we would be the same consortium if we didn't have that type of bridge with the biotech companies that we do that is so strong. And of course, our funding to recognize NIH. So thank you. I'll be happy to take any questions, and thank you for your attention.